Quickly here, as I said, I want to skim through these couple of slides, but point out the important ideas in these slides. Um, and if you're looking to have the reference or the slides, just communicate with me, because I have to meet the time of the lecture. Already spent some time with many concepts. This is reverse transcriptase DT-PCR. It is now we looked into the droplet digital-based PCR that can go and work with take the sample and, as we looked earlier, and partition that into many thousands of droplets. So this is a very interesting idea. And once combined with reverse transcriptase, we start thinking about quantification and gene quantification of what? Of gene transcripts. So you can look here at the two-step QRT-PCR and then one-step RT, reverse transcriptase, DT-PCR, and see some of the differences here. But the very interesting application here is single cell application. Perhaps you have heard about single cell applications. Think about, we have an eye tissue, and then we have, we are interested in knowing the gene expression in these different types of cells. We have many cells in specific tissue of an organ. And they want to see what makes gene transcription unique between the different sites at the granularity of the single cell. So instead of having one sample from the organ, we'll want to focus more and zoom in more and start looking into one single cell. And this can match with the idea of a droplet with that granularity. So DT-PCR or RT, these kinds of technologies can be used to bring us single cell technologies. At the end, there will be some nice, interesting visualizations, as you can see here, where we can cluster and experiment with some types of different cells and see if there's any different behavior or pattern. And this also brings this idea of single cell encapsulation. Start looking at the counting number of positives or negatives when there are two different genes, target and reference gene, in different cells. Eventually, we have thousands of cells that we can screen and see the output to compare and look into different, do a comparison of different experiments. So what I want to do here is show you, I'm gonna stop share and look into sharing my web view here. I'm gonna hopefully show you a website. Okay, let's look into this website. Oops, sorry. Bear with me. Okay. Okay, so this is a website I made during my own research as a postdoc in McGill, and part of that is what's called EcoTox chip analysis. So I want to show you this interesting visualization. I'm going to try an example data set. We'll visualize this. This is a qPCR type of analysis. We have specific species with different number of samples, 5 control samples, medium and high dose, meaning the chloropyrifos exposure study for the EcoTox chip, Japanese quail in this case. So anyway, I would recommend or advise you go and play with this website. This is hopefully now brings some flavor of, yes, bioinformatics analysis. Still in this course, we are hoping gradually going to dive into more and more programming. This is a visualization for the chip. I myself made this visualization. So all what you see is CT values. As we put the CT low and high values, we can see all the range. However, if we are going to limit this, we can explore which specific genes meet our own constraints. So we have gene XDH, for example, and we can read the CT value here. Now, this would be subject to further analysis. If we go up You can see we have some housekeeping genes. There's delta C, RTC, PPC values. So we're trying to take the RTC values and PPC values, subtract that from RTC values. So again, this is kind of different ways to try to measure and see if there's any issues in terms of the quality. So anyway, I'm just going to invite you to try to look into this and experience with it. It will take you through very interesting view, as well as if I go to the next steps, I'll be able to see some kind of interesting visualizations, as you see here. I'm going to run an experiment. This whole web-based tool is based on the back end on R programming, front end Java, to try to make it very easy for the user, all the way until we get into statistical analysis. Hopefully something we'll learn about more in the future. And then we'll be able to see differential expression analysis and see these based on the qPCR analysis, which genes are showing very interesting pattern in terms of a change of values across the control, high and medium in this case. Like this gene specifically here shows more response on the high dose as compared to the other classes or cases. Okay, maybe something we'll dive more into in the future. For now, I'll go back to the slides and get into interesting discussion on what is called graphical abstracts. Graphical abstracts is this idea that as you look into different research papers, we are hoping to find a good summary for this whole paper. It is becoming a common practice to develop a graphical abstract for the research paper, something you may want to think about as you will develop your own project of having one figure that can summarize information. In the office hour the other day, we have looked into an example of some research paper and we checked the graphical abstract there. In fact, during the presentations of the different lectures, several graphical abstracts from different papers were used to introduce you to some of the thoughts and ideas. When we look into the graphical abstract, of course, it is a decision that we need to, or several decisions that we need to make. How much that is specialized, giving the right audience. Is this for the target audience or not? What is the background of the target audience? And especially when it comes to fields like bioinformatics, you can be talking to scientists with more of a programming computer science background, and in some other venues, in one venue and another venue, it could be more of a biology and chemistry background. So this decides on what kind of information you want to put in the graphical abstract. So if you're trying to publish your work, in other words, are they looking for more biotechnology contribution, more on the algorithm side contribution that is going to highlight what kind of information we have. So I'm going to show you this graphical abstract as a third example. And again, those are not meant so that I can go, I need to go and explain them, just to give you certain ideas here in these different examples, how they are organized differently, showing different parts, explaining different ideas. But this is one example here that I want you to think. Do you like this graphical abstract? Think about it. And this is the last treasure hunt question. What kind of suggestions would you suggest to improve this graphical abstract? You might think about the amount of text here, the white spacing, the different organization arrangement. It can look kind of interesting poster that we can have in some scientific conference and so on and so forth. There are different tools that can be used, very nice. So BioRender here. It comes with lots of objects. You see all of these fancy images that sometimes you kind of think, oh, how did they do this? So before my own experience, I used to meet people where they would have to get experts or be trained to use Adobe Photoshop tools, for example, to make all of these interesting drawings. Now there are different tools that you can think of to draw all these scientific kind of very nice abstract. So think about using any of those. BioRender comes with the free version that gives you the chance to generate few figures, I guess 5 figures, and something you can have in terms of presenting your own work and your own project. So I hope you enjoyed this lecture. And again, there are still lots of biology concepts. It's a good way to establish our thinking. We started with the molecular biology side. We built up into a different layer right from the beginning, started looking into how we can move from a gene to a bigger picture, understand translation, transcription. And now, at least what you capture from this module is that there are technologies, many technologies, in fact, and they can do a lot for us. And this would mean there are going to be lots and lots of data. The idea is how we can use this simple idea of that toy ball that we have, the green fluorescence toy ball, that when is impacted by light, it have an energy that when we turn the room into dark light, we see that fascinating greenish glowing color that is going to help us to quantify many of the things. So I hope you enjoyed this, and see you in the coming office hour.